Hello, I'm Dr. Aizaz from MedicoVisual.com, and in today's visual lecture we will talk about the development of spinal cord. So in the last lecture we had seen that here there is this neural tube, and we had seen that if we cut a section of neural tube here, that this neural tube, it developed basically three stratas or three zones or three layers. So innermost is the ependymal zone or ventricular zone, which is nearest to the lumen of neural tube. Then outer to it is the intermediate zone or mantle zone, and then outermost is the marginal zone. Now basically it consists of, if we talk about the ependymal zone, it consists of this type of epithelial cells, columnar epithelial cell, which were forming the ependyma like this. Ependymal epithelium is here, but for the sake of simplicity I have just drawn it in a very simple way, right? Then in the mantle zone or intermediate zone we had the cell bodies and dendrites of your beloved neurons. So it basically consists of cell bodies and dendrites of neurons basically. And of course in the marginal zone consists of basically the axons. So marginal zones consist of axons. Marginal zone is white matter, and the mantle zone or intermediate zone is basically the gray matter. All these things we have discussed them previously in the lecture of histogenesis of neural tube. If you haven't watched that lecture, please watch that first to understand these things. Now if we move forward, of course also we have the pia matter surrounding it from the outside. Now if we move forward, that what happens that the mantle zone or intermediate zone of this neural tube in the region of spinal cord basically. So here there will be formation of spinal cord in this region, basically the caudal part of neural tube that will form the spinal cord. So in the future spinal cord region of the neural tube, the mantle zone is rearranged and reorganized basically in such a way that we have this type of orientation, and we have these dorsal parts here, these dorsal part here, and these are the ventral part. The ventral part, these are called basal plate. So this is basically nothing more than the rearrangement or reorganization of the mantle layer or mantle zone. So here we have the formation of basal plate, and here we have the formation of alar plate or alar plate. Then we have this shallow groove here. You can see we have this shallow groove in the lateral wall of neural tube in this region. So this is called sulcus limitans. Basically sulcus limitans demarcates the alar plate. It demarcates the alar plate from basal plate. That basically at this site here the alar plate ends, and if we go ventrally the basal plate starts. Here as well alar plate ends here at the site of sulcus limitans. This is the demarcation, and then the basal plate starts if we go ventrally. From basally we are going ventrally like this. So that's how the things are organized. Of course we have the similar marginal zone, and of course outside that we have the pia matter. And here we have this roof plate. This part is actually the roof plate, and this part is actually the floor plate, right? Now it will be reorganized further, and ultimately the alar plate, this is the alar plate, this is the basal plate. Basically alar plate will form the sensory component and basal plate will form the motor component. Now what will happen that the alar plate will be converted into dorsal gray column. The basal plate, it will be organized into ventral gray column. Now this dorsal gray column, it is associated with the sensory component. It receives the sensation from your body, and the ventral gray column, it actually gives the motor information towards the muscles and other components. And then we have formation of ventral median fissure here, and this dorsal median septum at the dorsal side. Now in the region of the thoracic and upper lumbar region, We also have the lateral column. So lateral column is associated with the autonomic nervous system. We will not go into details in this lecture. We have already discussed these things in another lecture that I will put the link in description. Now, as I've told you that dorsal gray column is the sensory, it is afferent. It means that it will later we will see that it receives the sensation. So it will receive the sensations like this. So there will be the formation of dorsal root ganglia, and from there the nerve will be formed, and this nerve will bring the sensory information to neurons in the dorsal gray column. And then we will also see that at the site of ventral gray column, the axons will arise from these neurons, and they will go to muscles or other motor organs, and they will help in locomotion of these muscles and so on. Now, how can you remember that Alar plate or dorsal gray column is associated with sensory information, and the ventral gray column or basal plate, which is actually derived from basal plate, how can you remember that it is associated with motor information? Dr. Najib has devised a beautiful visual mnemonic for this. So here is the mnemonic. So let's suppose if we draw something like this. Actually, Dr. Najib originally drew a rabbit shaped thing, but here, because I love cats, so I have drawn a cat, although I know it's not a beautiful cat. Anyhow, so these are the ears of the cat, and ears represent the sensory component, and it is the basal, not basal, it is the Alar plate, or it is the sensory. Ears are sensory, so this is the Alar plate. And then here we have these legs are basically the denoting the motor component. So these are the basal plates. So if you draw a diagram like this, you can remember this. Hats off to Dr. Najib for coming up with this beautiful mnemonic. It's not my creation. Full credit goes to Dr. Najib for this. So let's move forward and let's see the 3D model of the spinal cord. So here you can see we have this lateral column. So as you can see that the lateral gray column, it does not reach all the way towards the end of the spinal cord. It starts actually from T1, thoracic 1, and it reaches up to about L2 spinal level like this. And then of course we have here is the basal plate derived ventral gray column. You can see clearly in this 3D model that it is a column shaped, a properly formed column. And then we have the dorsal gray column, which is derived from which plate? It is derived from Alar plate. So Alar plate derived dorsal gray column, and basal plate derived ventral gray column. And of course we have the lateral gray column. And by the way, please note that the lateral gray column is also the subdivision of basal plate. Actually, it is the component of basal plate. You can say it is also derived from basal plate. So you can see that it is present on the other side as well. If we rotate it like this, it is present on the other side as well. So this was the beautiful 3D model of the spinal cord. Of course, it's the ventral median fissure. And then here we are back with the 2D model, 2D diagram. And as I've told you, basically it consists of the ventral gray column. It consists of lots of cell bodies of lots of neurons, lots of motor neurons. And here I have shown only a single motor neuron. So this motor neurons, its axon will come out of this, of course, and it will be forming the motor component, motor ventral root, basically ventral root of the spinal nerve. And here then what happens that we have neural crest cells, and we have discussed already that they arise from the crest of neural tube. When neural tube was developing, some cells at the crest, they separated and converted into mesenchymal cells, and those cells, they form the neural crest cells. And these neural crest cells, they form many different types of cells, but one of the important types of cells that they form are these axons in this region. They will form special type of neurons, which initially are of course apolar, then they form the two cellular processes, like two cellular axons basically, and become bipolar neurons. Then what happens that these bipolar neurons, they will be converted into something like this. So now this is interesting situation that now this neuron, now it has a single process actually. So here we have this neuron has a single process like this, but this single process, it divides into two axon like processes. So it is actually unipolar neuron. It has a single pole. It is actually unipolar neuron, but it will act as a bipolar neuron. So that's why we call it pseudo unipolar neuron. Let me explain this thing. So if we move forward, so what will happen that one of its component will go here, and here it might synapse with another neuron here, or it may go to the dorsal column track and go upward. We will discuss those details in neuroanatomy. It will form the dorsal column medial lemniscus system, and this other peripheral component, it will go to some sensory organ. For example, it might go to, let's suppose, skin. It might go to skin, and here, for example, if someone pricks you with a pin, then the pain sensation will go through this, and this peripheral division of this unipolar neuron. This is unipolar neuron, but its peripheral division, it will bring the sensation towards the cell body here, and then this central division, it will take sensation away from the cell body towards the spinal cord. So basically this axon like process, it will act as a dendrite, and this will actually act as a axon. So actually it's a unipolar, but functionally it's a bipolar. This part is acting as dendrite, this is acting as axon. So functionally it's a bipolar, but structurally it's a unipolar. So that type of neuron is termed as pseudo unipolar neuron. So a lots of pseudo unipolar neuron will form here like this. Their cell body will join here, remain here, their cell bodies will remain here, and their cell body will clump here. They will sort of gather here, and their cell bodies here will form a ganglia, and this ganglia is called dorsal root ganglia. And of course this ganglia is actually formed by neural crest cells, indirectly formed by neural crest cells, because these neural crest cells will form these pseudo unipolar neurons, and this pseudo unipolar neuron will be actually forming this dorsal root ganglia. Here is basically this part, this is actually rootlet. This root is formed, this rootlet is called dorsal root. This is gray column, dorsal gray column. It's not root, it's a column, dorsal gray column. As I've shown you in the 3D model previously, it's a gray column. And this is the root. It's a rootlet or root that is originating here. And here this is at the ventral side, of course. So it's a ventral root. So here is the ventral root. Ventral root is originating from ventral gray column. So it's a ventral root which originates from ventral root of spinal nerve, basically of spinal nerve. It is also of spinal nerve. And this is the dorsal gray column of spinal cord. It's a ventral gray column of spinal cord. It's a ventral root of spinal nerve. It's a dorsal root of spinal nerve. Then of course here is this ganglia will be formed here, which is the dorsal root ganglia. Then what happens that the dorsal root as well as ventral root of spinal cord, these both roots of spinal cord, they will combine to form a single spinal nerve here. So this is a single spinal nerve. It will form a single spinal nerve. This is a proper full-fledged spinal nerve. And if you look carefully, this spinal nerve, it consists of the dorsal component, which is providing with the neuron, with the sensory neuron, and ventral component, ventral root, which has provided it with the motor neuron, with the efferent neuron. This has provided with afferent neuron, this has provided with efferent neuron. So this spinal nerve, it has sensory fibers as well as the motor fibers. Both type of fibers are there in the spinal nerve. Then what happens that the spinal root, it will be divided into two parts. This is the ventral rami, dorsal rami, sorry, this one is the dorsal rami, dorsal ramus or dorsal division. Basically it's a dorsal division, dorsal ramus of spinal nerve, and this is the ventral ramus of spinal nerve, ventral ramus. This ventral division will supply the interior body wall, the chest wall or the abdominal wall, and the muscles of the limb and so on, of the skin, of course, of that region. And dorsal ramus, it will innervate the back of the body. Let me show you with another diagram, another sketch. And of course, please note that all these things are bilateral. Here also there will be the dorsal root of spinal nerve, here there will be ventral root, and so on. All these things are bilateral. I have just shown on one side only, but please remember it's a bilateral, right? So let me show you all these things in another sketch so that you really understand it properly. So here you can see again that here is the dorsal root of spinal cord, here is the ventral root. They both combine here to form the spinal nerve, and then this is the dorsal ramus, and here is the ventral ramus. And you can see that the dorsal ramus, it is supplying the muscles of back and of course skin of the back region and vertebral column and so on. And then you can see whole this is the ventral ramus, here whole this is the ventral ramus of spinal nerve, here also whole this is ventral ramus. Of course everything is bilateral. And you know that the ventral ramus as well as dorsal ramus, both of the rami of spinal nerve, they consist of sensory as well as motor nerve fibers. Both of the type of nerve fibers are there, and of course they are supplying the muscles of anterior body wall, abdominal or chest wall, and they are also supplying the limb buds here, developing limb buds here, which later will form the muscles of the legs and of course the arms as well. So this is the basic idea of spinal nerve. Now here there is another problem, that the conduction velocity, the nerve conduction velocity of such unmyelinated fibers is very slow. But opposite to that, the myelinated fibers, their conduction velocity is very fast. The nerve impulse jumps from node to node like this. So these are the nodes of Ranvier, one node, second node, and so on. We have discussed in detail that how this process occurs, what is the concept of saltatory conduction in another lecture. You can watch that lecture to understand its physiology, but we are not going into physiology. Now in embryology, we want to understand that how the myelination of nerve fibers occur. So basically we have the neural crest cells. They will form the Schwann cells, and Schwann cells will actually myelinate the axons of peripheral nervous system. In the central nervous system, we have neuroepithelial cells which will form the radial glial cells, and those will form oligodendrocytes. Initially, first there is the formation of glioblast, which will form the oligodendroblast, which will form oligodendrocytes, and then these oligodendrocytes will then myelinate the axons in central nervous system. So Schwann cell will myelinate axons of peripheral nervous system, but oligodendrocytes will myelinate the axons in central nervous system. So first of all, what will happen, let me explain it on the sketch. That let's suppose here is a nerve fiber, So here is, let's suppose, a nerve fiber. So first of all, what will happen that some Schwann cells will come here. Some Schwann cells are there. They will come here, and they will surround this. They will surround it. They will surround this neuron from all side like this. They will surround it, and this neuron as well. They will sort of hug this neuron like this. Then what will happen that they will sort of roll their cell membrane around this neuron. They will sort of roll this their cell membrane several times around this neuron to form a sheath, neuralimal sheath or myelin sheath. So that's how the myelin forms. Let me show you the animation so that you clearly understand that what is going on here. So let's suppose lots of Schwann cells have surrounded this neuron. You can see here they have surrounded this neuron. I have deliberately made them a bit translucent so you can understand what actually is going on here. Now if we watch the animation, you can see that their cell membrane will roll around at the specific regions of this nerve fiber, of this axon. Basically, you can see here. So they have rolled around, and in this way they have formed these neuralimal sheaths or myelin sheaths. So one Schwann cell, basically it can form the neuralimal sheath along a very small part of an axon. It cannot form the neuralimal sheath of whole of the axon. Second thing is that it will completely surround that portion, and it itself will become the component of this neural sheath. So you see, it's a living cell actually. So here the nucleus is there, so it itself will become the component of that myelin sheath. So it has been integrated into this myelin sheath like this. So we have several different Schwann cells forming the neural sheath of a single neuron. And of course we have these nodes where there is no Schwann cell, and nodes, the action potential can jump here like this. That's what they say, they jump from node to node, and we have discussed the details already. So this was the concept of myelination in peripheral nervous system. The myelination in central nervous system is exactly like this, that again there is the formation of, there is rolling up of the cell membrane of the oligodendrocyte. In that case, their processes, their cellular cell membrane processes, they will again roll up around the axon, but there are some differences. So let's see the differences. So the difference here in central nervous system is that the oligodendrocyte, a single oligodendrocyte, has many processes, many cellular processes, and a single oligodendrocyte, it can myelinate parts of up to 50 axons of 50 different neurons actually. So it's very unique type of configuration that a single oligodendrocyte, it can myelinate parts of up to 50 neurons, and oligodendrocyte, it itself does not become the part of that myelin sheath. It itself is not integrated into myelin sheath. It's like a hydra, you know, that Greek god Hydra who had lots of heads, and if we cut one head, it developed another head. So it's just like that it has lots of heads, I mean lots of cellular processes, and with those cellular processes, it can roll those cellular processes around the neuron, and it can again with those cellular processes, it can form the myelin sheath. So for example, here is the axon. So here is an oligodendrocyte. Let's draw it with another color. So let's make it pink. So here is, let's suppose, this cellular process. So this cellular process, it will roll around like this, similar to what happened in the peripheral nervous system in case of Schwann cell, and then it can roll around another one as well, another neuron as well like this. So here another neuron is there, and so on. It can roll around its process around many different neurons. So these are the oligodendrocyte. Previously we had discussed the Schwann cells in case of the peripheral nervous system. Now we will briefly discuss the positional changes in the spinal cord. So here is the initial configuration, that here is this spinal cord. So initially what happens, that let's suppose this is T1 vertebral column and this is T2 vertebral column. Initially this spinal nerve is T1. The spinal nerve originating between this is T1, and then this one is T3, and this one is T2, and so on. Last one is coccygeal nerve, and so on. The second last is S5 nerve, and this is S5, for example. So basically the spinal nerves are originating at the same vertebral level, but later what happens, that the vertebral column, they become more enlarged, they grow much more as compared to spinal cord. So vertebral column will become longer as compared to the spinal cord. So what will happen, that let me show you the animation. So there will be this diagonal arrangement like this. So spinal nerves will originate diagonally, right? So especially at the lower levels, the spinal nerve, they will originate diagonally from the spinal cord. So here in this diagram you can see clearly. So what happened here actually, that there is this diagonal arrangement like this. So for example, let's suppose this is, let's say, S2 here and it is S3 here. So this is the S2 here, but its origin, if we trace its origin, its spinal origin will be somewhat here. So its spinal origin is higher as compared to its vertebral level. So vertebral level is lower as compared to the origin of the spinal cord, origin of this nerve from the spinal cord. So they will originate sort of diagonally like this. The second thing that happened, let me show you the animation again. So the second thing that happened here, that basically the spinal cord is covered by meninges. 3 meningeal layers are there, and let me show you these. So we have these 3 meningeal layers. The innermost is pia mater. This is the pia mater, this purplish one here. Then outer one, this pinkish one, is arachnoid mater. Arachnoid mater. And the outermost is dura mater, which is nearest to bone. Dura mater. And between the arachnoid and pia there is this space called subarachnoid space. Here is this space, subarachnoid space, which consists of CSF. Now initially these meninges, they are somewhat close to each other, and the pia mater, it has special affection with the spinal cord. It almost remains attached with, almost always remain attached with the spinal cord. But what happens, that when the spinal cord ceases to grow further and the vertebral column continue to grow, what will happen, that this pia mater, it will sort of form a ligament like structure. You can see here. So it will form a ligament like structure like this purplish one, ligament like structure. And about the arachnoid mater and dura mater, they will initially remain close to the bone. Initially they will remain close to vertebral column, but when they realize that the spinal cord is not coming with them, ultimately they will also come and meet with this ligament like structure that was formed by this pia mater. At S2 level, at S2 vertebral level, they will meet this ligament like structure. This name of this ligament like structure, which is initially formed by the pia mater, it is called Filum terminale. It is called Filum. Let me write it here. It is called this one. This is this one. It is called Filum terminale. It ultimately attaches with coccygeal bone of your vertebral column. So, and it anchors the spinal cord. It is called Filum terminale. So initially it is made up of this pia mater, but here is the S2 vertebra. here is the S2 vertebra. Below the S2 vertebra, when the dura and arachnoid, they also cover it from outside. Below the S2 level, the phylum Terminale, it is also covered by dura and arachnoid matter. By the way, in textbooks, they write about the dura matter, but most of the authors, they simply ignore the arachnoid matter. I don't know why this racist behavior with the arachnoid matter. I think the arachnoid matter should sue the authors of these textbooks for the discriminatory practices. They are discriminating against arachnoid matter. It is not even. But anyhow, thanks to Mr. Keith L. Moore, who has actually written the name of arachnoid matter as well, who has not ignored the arachnoid matter. Keith Moore and his co-authors. A special thanks to them for mentioning the arachnoid matter. Anyhow, so here let me tell you that the dura matter, initially it is sort of dura and arachnoid matter. Initially it is forming the a covering around the bone, around the vertebral column like this, and a sac is formed here. This sac is basically filled with CSF. In the newborn babies, it terminates at L3 level. This is L3. So initially it terminates the spinal cord, it terminates at L3 level, and in adults it becomes further higher. It terminates at about L1 to L2 level, mostly between L1 and L2. Below the L4 and L5, between L4 and L5, it is safe to insert a needle. So here you can insert a needle somewhat safely. You can still hurt these nerves, but somewhat safely you can insert the needle here for the lumbar puncture, and you can take the cerebrospinal fluid for testing purposes. You can send it in the lab for testing purposes. So that's why the lumbar puncture is usually done between the L4 and L5 vertebral column level. Finally, a few things that I would like to talk about here. Basically, there are three terms that are often confused by medical students. What are those three terms? One is phylum Terminale, and then there is a term called Cauda Equina, and then there is a term called Conus Medullaris. What are these three terms? Let's talk about it one by one. Now here you can see that spinal cord terminate here at this level, at L3 level in newborn babies, and L1 or L2 level in adults. So at this level you can see the spinal cord is initially, it's a column, it's a you can say cylindrical structure. Initially it's a cylindrical structure, but near its termination it forms a cone-like structure like this. It narrows down and it forms a cone-like structure. Here. This terminal ending cone of the spinal cord, it's the end of spinal cord at L3 level in newborn, at L1 or L2 level in adults. This cone-like structure here, this cone-like structure which demarcates the ending of spinal cord, this is actually conus medullaris. This is conus medullaris. Then if you come below the conus medullaris, if you go caudal to the conus medullaris, you can see that here there is no spinal cord because spinal cord has terminated here, but you will see these nerves here, these nerve roots here. So these nerve roots, they give an appearance of the tail of a horse. So scientists, anatomists have put a very artistic name. They call it horse tail, or in, I think, Latin it's called Cauda equina. Equina is horse, Cauda is tail. So they call these nerves that are below the conus medullaris, these nerves which give an appearance of horse tail, they call it Cauda equina. So Cauda equina is these nerves which are below the level of conus medullaris, which are below the level of termination of spinal cord. So these are Cauda equina. Conus medullaris is something else, Cauda equina is something else. You must be very clear between the differences between two. And finally, Filum terminale, we have discussed already that it's a ligament which is initially formed by pia mater. Not initially, I should say, one of its component, its cranial component, it is formed only by pia mater, but as this subarachnoid sac terminates at S2 level, here is the S2 level, as it terminates at S2 level, so this subarachnoid, this arachnoid matter as well as pia mater, both of them, they join it like this. And of course we have pia mater as well. So pia mater, not pia mater, I mean dura mater, it will also join here. And this caudal part of Filum terminale, it is made up of all three meninges. Internally we have pia mater, then we have arachnoid, and then we have dura mater. So this is Filum terminale externa, this is Filum terminale interna, and it anchors the spinal cord. It is downward, it is attached with coccyx bone. So it anchors the spinal cord with the coccyx bone. So this was the Filum terminale. So you must know clearly the differences between two. Of course, we will discuss its detail in the neuroanatomy as well, but you must have some basic idea at this time. So that was about the development of spinal cord. Thank you so much for watching this video.